Every macromolecule in every cell in your body is built using the same principle a 5-year-old uses with a box of Lego bricks. Small standardized pieces snap together to build something bigger and more useful than any single piece could ever be. AP Biology calls those small pieces monomers, and the assembled structures polymers. And once you see that Lego logic, half of Unit 1 Chemistry of Life turns into pattern recognition. A monomer is one small molecular building block. A polymer is a long chain built from many monomers linked together. The size difference is real, but the bigger insight is functional. Individual monomers are simple, while polymers can store energy, catalyze reactions, build membranes, or carry genetic information depending entirely on how those identical bricks are arranged. Same monomer, different polymer, completely different cellular job. 4 pairs come up on almost every Unit 1 question, and you should be able to recite them in your sleep. Carbohydrates use monosaccharides like glucose as monomers and form polysaccharides like starch and cellulose. Proteins use amino acids as monomers and form polypeptides. Nucleic acids use nucleotides as monomers and form DNA and RNA. Lipids are the exception we will come back to. Memorize those 3 classic pairs, and you handle most of the multiple-choice questions on this topic. Polymers do not assemble themselves. 2 reactions build and break them, and the AP exam tests this distinction relentlessly. Dehydration synthesis removes a water molecule between 2 monomers so a new covalent bond can form, which builds the polymer one link at a time. Hydrolysis adds a water molecule to break a bond between monomers, which splits the polymer back into reusable pieces. The memory hook is direction. Removing water builds. Adding water breaks. Here is the structure function payoff that scores on FRQs. Starch in this pasta and cellulose in plant cell walls are both polymers of glucose, the exact same monomer, but they do completely different jobs because the bonds between monomers are arranged differently. Starch branches and dissolves to release glucose for energy, while cellulose forms straight rigid fibers for structure. Same brick, different blueprint, different function. Always cite that logic when comparing related polymers. The bond-linking monomers tells you which macromolecule class you are looking at. Carbohydrate monomers join through glycosidic bonds. Amino acids join through peptide bonds, which give a protein like the hemoglobin shown here, both its sequence and its shape. Nucleotides join through phosphodiester bonds in the sugar-phosphate backbone of DNA and RNA. If an AP stem mentions one of those bond names, you immediately know which macromolecule the question is testing. Lipids are the most tested exception in Unit 1, so do not skip this. They are macromolecules because they are large biological molecules, but they are not classic polymers because they are not built from many identical repeating monomers. A triglyceride is one glycerol attached to 3 fatty acids, while a phospholipid has 1 glycerol, 2 fatty acid tails, and a phosphate head. The safe AP wording is that lipids are macromolecules grouped by hydrophobic behavior, not by repeating monomer chains. The 5-point move on any monomers and polymers, FRQ, is always the same 4-part structure. Name the monomer. Name the bond that links them.
You can drill 18 flashcards and 14 AP-style practice questions on monomers and polymers in under 15 minutes using the link below. That is the difference between a 3 and a 5. Subscribe for new AP videos every week.